Hey everyone, ready for a deep dive into something pretty amazing?
I know I am.
Well, we're going to unlock some secrets today about molecules and reactions and all that good stuff in the world of organic chemistry.
Definitely more exciting than it sounds when you read about it in a textbook, right?
Oh, yeah, for sure. And especially today, I think, we're going to go deep into like the core principles of organic chemistry. So we're talking carbon-containing compounds, really.
All about carbon.
All about carbon. Okay. Well, I'm excited to get into it. So are we going to find out like why carbon is so special?
Yeah, so that's the first thing, is it's a superstar element. And
Yeah.
and really what makes it so special is its ability to form these long chains and rings. So you can imagine like
Oh,
carbon
uh-oh.
atoms, like these tiny little Lego blocks that can just keep connecting and connecting.
That's a good visual. I like that.
Yeah, that makes them the backbone of like literally millions of compounds.
Wow. I mean, so we're talking everything from the gas in our cars to like the proteins in our bodies.
Yeah. Exactly. I mean, even the DNA that makes up, who you are.
That's wild. Okay, so but wasn't there this idea like back in the day that organic compounds could only come from like living things?
Yeah, they used to think that organic compounds were only made by living organisms because of this like mysterious vital force,
Oh, wow.
right? Like there was something special about living things
Yeah.
that could make these compounds, but then came along a chemist named Friedrich Wöhler in 1828. He did this pretty groundbreaking experiment.
What'd he do?
He accidentally synthesized urea, which is
Oh.
an organic compound found in urine, from an inorganic salt.
Wow. Okay, so that basically debunked that whole idea, huh?
Exactly. It kind of like shattered that myth of the vital force, and it
Yeah.
really launched this whole modern era of organic chemistry.
Wow, that's so cool. Okay, so we know that like carbon can form these chains and rings, but how do those actually like look in 3D? I mean, are they just flat or what's going on there?
Great question, because molecules, they're not just, flat drawings on a piece of paper, right?
Right.
They exist in three dimensions. And understanding their shapes is super important for understanding their properties
Yeah.
and how they interact with each other.
Yeah.
And that's where we introduce the concept of hybridization.
Hybridization. Okay. I'm intrigued.
So, carbon's electrons are arranged in orbitals, those regions around the nucleus, and these orbitals can actually combine and rearrange themselves
Oh, interesting.
to form hybrid orbitals, and that influences the shape of the molecule.
Okay, so it's kind of like the electrons are doing a little dance,
Yeah.
changing the overall shape of the molecule.
Yeah, that's a
Yeah.
great way to put it.
Cool.
And there's three main types of hybridization that carbon can undergo. So you have sp3, sp2, and sp.
Okay, CP3, sp2, sp. So each of those leads to a different shape.
Exactly. Each one kind of dictates a distinct molecular shape,
Got it.
determines how those atoms are arranged in 3D space.
Gotcha. Okay, well, let's get into specifics. What's P3 all about?
Let's start with sp3. And the best example here is methane, the simplest hydrocarbon.
Yeah, that's it.
One carbon atom, four hydrogen atoms. In methane, that carbon uses sp3 hybridization. So what that means is that one s orbitals and three p orbitals from the carbon, they combine to form four identical sp3 hybrid orbitals.
Okay, so we're mixing and matching those orbitals. What does that do for the shape?
Well, these four sp3 hybrid orbitals, they point towards the corners of a tetrahedron.
Tetrahedron.
So imagine like a pyramid, but with a triangular base.
Oh, okay.
And the carbon atom sits right in the center of this pyramid, and the four hydrogen atoms are at the corners.
Got it. So it's like in the middle of this pyramid, the carbon is and the hydrogens are at the points. Pretty stable structure, right?
Very stable.
Cool. Okay. What about sp2?
Okay, for sp2, let's think about ethene.
Ethene.
So that's a molecule with a carbon-carbon double bond.
Okay.
In ethene, each carbon uses sp2 hybridization. So this time, one s orbital and two p orbitals combine to form three identical sp2 hybrid orbitals.
Okay, so we're leaving one p orbital kind of out of the mix this time.
Right, and that one remains unhybridized.
Okay. How does that change things?
Well, these three sp2 hybrid orbitals, they arrange themselves in a plane kind of pointing to the corners of a triangle.
Okay, got it.
So this gives us a flat, planar structure for ethene.
Planar.
The two carbon atoms are connected by that double bond, right?
Yeah.
And that double bond is made up of one sigma bond, which is formed by the overlap of two sp2 hybrid orbitals, and one pi bond, which is formed by those unhybridized p orbitals.
Okay, so the pi bond is from the unhybridized ones. The sigma is from the hybrid.
Okay.
Got it. So we've got a tetrahedron. We've got a flat plane. What about sp? What does that look like?
For sp, think of ethene.
Ethene.
That's a molecule with a carbon-carbon triple bond. In ethene, each carbon uses sp hybridization, so one s orbital and one p orbital combine, forming two identical sp hybrid orbitals. And in this case, two p orbitals are left unhybridized.
Okay, so even fewer orbitals are mixing this time. What kind of shape do we get?
Well, those two sp hybrid orbitals, they arrange themselves linearly,
Linearly.
pointing in opposite directions.
Okay.
And this creates a linear structure for ethene. So the two carbon atoms are connected by that triple bond.
Okay.
And that triple bond consists of one sigma bond from those sp hybrid orbitals and two pi bonds from the unhybridized p orbitals.
Okay, so a triple bond has two pi bonds and one sigma.
Got it. So we've got our tetrahedrons, our flat planes, and our straight lines.
Exactly.
It's kind of wild how these little changes can make such different shapes.
It is.
What about double and triple bonds? Those seem to be more like reactive than the single bonds. Is that true?
Absolutely, yeah.
Okay, so why is that?
Well, the double and triple bonds, they contain those pi bonds, right?
Yeah.
And those are formed by the sideways overlap of those unhybridized p orbitals.
Now, pi bonds are actually weaker than sigma bonds, which are like that head-on overlap.
Oh.
But their weakness actually makes them more susceptible to reactions.
So it's almost like they're waving a flag, like, come react with me.
Yeah, exactly. It's almost like they're like holding up a sign saying, I'm ready to react.
Yeah, exactly.
Yeah.
Okay, that's interesting. So are there other interesting things about those pi bonds?
Yeah. I mean, one of the things about the pi bonds is that they actually restrict rotation around the carbon double or triple bond.
Wait, restrict rotation. What do you mean?
So imagine you've got like two groups of atoms attached to a carbon double bond. Because of those pi bonds, those two groups, they can't just freely spin around like the double bond axis,
Oh, okay.
like they could with a single bond.
Gotcha.
They're kind of locked in place
Oh, interesting. Okay.
relative to each other.
Okay, so that restricted rotation, is that important for anything?
It's incredibly important because it actually influences the properties and reactions of so many organic compounds. Okay, it's one of the reasons why organic chemistry is so diverse.
That's so cool.
Even small changes in the molecule shape, it can have a huge impact on how it acts.
Yeah, that makes sense.
Huh.
Okay, well, all this is really fascinating, but I got to say, picturing these like complex 3D structures in my head is kind of tough.
Yeah, I can imagine.
Like, how do chemists even keep track of all the atoms and bonds, especially when these molecules get like huge and complicated?
Yeah, well, chemists, we've developed all sorts of ways to represent these molecules.
Oh.
We have detailed structural formulas where every single bond is shown.
Okay.
But then we have simplified line structures where, carbon and hydrogen atoms are implied.
So it's kind of like you've developed this like secret code to make things simpler.
Yeah, exactly. Like a shorthand for chemists.
That's cool. Can you give me like an example of that?
Sure. Imagine butane.
Butane, okay.
It has 4 carbon atoms in a chain. The structural formula would show every carbon, every hydrogen, every bond connecting them.
Yeah.
But its line structure would just show a zigzag line with 4 vertices.
Okay.
And each vertex represents a carbon atom.
Ah, okay. So it's like this minimalist drawing, right? Like just the bare bones of the molecule?
Yeah, that's a good way to put it.
That's cool. But how do you like show the 3D part? Like those tetrahedrons and planes we talked about.
Right. Well, to show the 3D arrangement of the atoms, we use something called the wedge and dash system.
Okay.
So solid wedges, they show bonds popping out of the page towards you.
Okay.
Dash wedges mean bonds going away from you. And regular lines, they're just bonds lying flat in the plane of the page.
So it's like those pop-up books from when we were kids.
Yeah.
Adding that 3D element.
Exactly. Yeah. It's a simple but powerful system.
I like it. Okay,
Yeah.
so we've got our line structures, our wedges and dashes, but with like millions of organic compounds out there, how do you even begin to like categorize all of them?
That is the question, right?
It seems like it'd be so easy to get lost.
Yeah. Well, organizing this huge molecular world, it's really essential for understanding how these compounds relate to each other and predicting their properties and reactions.
Yeah.
So we have to classify them.
Classification. Okay, so it's kind of like, I don't know, like library card catalog, helping us find what we're looking for.
That's a great way to think about it, yeah.
Okay, cool. So
Well,
how do we classify these compounds?
one way is by their carbon skeleton.
Okay.
So you can ask, is it open chain, like a string of beads?
Okay.
Or is it cyclic, forming a ring?
Okay. Open chain versus rings. That's like pretty clear distinction.
Yeah, open chain compounds are also called aliphatic compounds.
Okay.
And you've already met a few of them: methane, ethane, butane.
Right, those simple hydrocarbons we were just talking about. What about cyclic ones? Any examples there?
So you've got cyclohexane, that's 6 carbon atoms in a ring.
Yeah.
And then there's benzene, which is 6 carbon atoms, but it also has those alternating double bonds.
Benzene. Yeah, that one's famous.
So special about benzene? Well, benzene and other aromatic compounds, they have unique properties because of those delocalized electrons in the ring.
Okay, interesting. So we've got our chains, our rings, our special aromatic rings. But are there other ways to, like, group these compounds?
Of course, yeah.
Okay, what else?
I mean, carbon skeletons are just one piece of the puzzle, right?
Right.
Another key way to classify them is by their functional groups.
Right, the molecular tags. They tell us how a molecule might interact with others.
Exactly. For example, the hydroxyl group, that's AOHO,
Right.
that's the hallmark of alcohols,
Right.
gives them their characteristic properties. Aldehydes, they have a carbonyl group, that's AAOCO, at the end of a carbon chain. Ketones, they have that carbonyl group, CAO, within the carbon chain.
Okay.
Carboxylic acids, they have that carboxyl group, AAOOH, and that's what makes them acidic.
Right. So it's kind of like those labels on, like, food packaging telling you what's inside.
That's a great analogy.
Okay, so how do we keep track of all these different functional groups? Seems like there's a ton.
Well, chemists, we always want to make sure we're speaking the same language, right?
Right.
So we've come up with a systematic way to name these compounds.
Okay.
It's called IUPAC nomenclature.
IUPAC nomenclature.
Hmm.
Sounds intense, but I guess it's like any new language. You learn the rules,
Yeah.
and then you can start communicating.
Exactly. And honestly, once you understand the basic principles, it's really not as hard as it sounds.
Okay, well, I'm up for the challenge. Let's start with, like, the simplest ones, the alkanes. How do we name those?
Okay, so with alkanes, the key is to find the longest continuous chain of carbon atoms.
Okay.
That's the parent chain.
Okay.
And its name is based on how many carbon atoms it has. So 1 carbon is methane, 2 carbons, ethane, 3 propane, and so on.
Okay, so far so good. But what if there are, like, branches coming off that main chain?
Great point, because real molecules, they're not always straight chains, are they? Those branches, they're called alkyl groups, and they're essentially just alkanes, but they've lost a hydrogen atom.
Okay,
So they have a bond available to attach to something else.
gotcha.
And we name them by replacing the ending of the alkane with GL. So methane, CH4, becomes methyl, CH3. Ethane, C2H6, becomes ethyl, C2H5.
So they're like the sidekicks ready to branch off and make things more complex.
Yeah, exactly. And when we name these branched alkanes, we got to specify where those alkyl groups are on the parent chain.
Okay, how do we do that?
We number the carbons in the parent chain,
Okay.
and we start from the end that gives the substituents, the alkyl groups, the lowest possible numbers.
Okay, so it's like a game of find the shortest route.
That's a good way to think about it.
Okay, so if you have a methyl group on the second carbon in a 4-carbon chain, would you call it 2-methylbutane?
Exactly. You got it.
Wow, okay. I'm getting this. What if we have, like, multiple alkyl groups? How do we handle that?
If you have multiple identical alkyl groups, then you use prefixes like D, tri, tetra
Yes.
to indicate how many there are.
Got it.
And if you have different alkyl groups, you just list them alphabetically.
Okay, so it's all about being super organized and systematic.
Yeah, we want to make sure that everyone understands exactly what molecule we're talking about.
Yeah, that makes sense. Okay, but what about those functional groups? Where do they fit into the name?
Well, usually when you have a functional group, it's kind of the VIP of the molecule.
Okay,
It takes precedence over alkyl groups in the name.
gotcha.
We use suffixes to show these functional groups. So alcohols get ol, aldehydes get ol, ketones get one, carboxylic acids, oic acid.
So it's like adding a special ending to the name to signal that functional group's there.
Exactly. And just like with branched alkanes, we use numbers to show where the functional group and any other substituents are on the chain.
Got it. Okay, so how would we name an alcohol with a hydroxyl group on, like, the 3rd carbon of a 5-carbon chain?
That would be pentanthreol.
Pentanthreol.
Gotcha.
Okay, breaking it down. Pentan because it's 5 carbons. 3 because the hydroxyl's on the 3rd carbon. And ol because it's an alcohol. Okay, how about an aldehyde with the carbonyl group on the second carbon of a 4-carbon chain?
That would be butan-2-al.
Butan-2-al.
Mm.
Okay. And last one, a carboxylic acid with the carboxyl group on the first carbon of a 3-carbon chain.
That's propanoic acid.
Propanoic acid. Okay, this is making sense. But what if there are multiple functional groups? Does it just get crazy?
Not at all.
Okay.
There are rules for those multifunctional compounds, too.
Oh, good.
We identify what's called the principal functional group.
Okay.
That's the one with the highest priority.
Okay.
And then we name it based on that.
Okay.
The other functional groups, we treat those as substituents.
So it's like they're fighting for the top spot in the name.
Exactly. It's like they're all competing to be the star of the show.
That's cool.
Yeah.
Okay. What if you have 2 molecules and they have the exact same molecular formula, so the same number and types of atoms, but they're arranged differently? Wouldn't they have different properties?
That's a really insightful observation.
Oh, okay, good.
That brings us to this idea of isomerism.
Isomerism, right.
Isomers are compounds with the same molecular formula but different structures. So that means different physical and chemical properties.
Okay, that makes
It's
sense.
like, having 2 Lego sets with the same pieces but building completely different things with them.
Yeah, totally.
Yeah.
So, like, what kinds of isomers are there? What are those different Lego creations?
Well, there's 2 main types. Structural isomerism, where the atoms are connected in a different order, and then there's stereoisomerism, where the atoms are connected in the same order, but they're arranged differently in space.
Okay,
So like if you had 2 Lego models and they were mirror images of each other.
got it. So let's start with structural isomers.
Yeah.
What are some examples of those?
So one type is called chain isomerism.
Okay.
That's where those carbon atoms are arranged in different chains.
Oh, yeah.
So butane and isobutane, they both have the formula C4H10, but butane has 4 carbons in a straight chain, while isobutane has a 3-carbon chain with a methyl group branch.
So it's like building a straight Lego tower versus one with a little side wing.
Exactly.
Cool. Okay, what else?
Then you have position isomerism, where a functional group or a substituent is attached to a different carbon.
Okay.
So like 1 propanol and 2 propanol, they both have the formula C3H80, but the hydroxyl group is attached to the first carbon in 1 propanol and the second carbon in 2 propanol.
So like moving that hydroxyl Lego piece to a different spot in the chain.
You got it.
Got it.
And then we have functional group isomerism.
Okay.
And that's where the atoms are arranged to form different functional groups.
Okay.
So like propanol and propanone, both with the formula C3H60.
Right.
Propanol has an aldehyde group, while propanone has a ketone group.
Oh, okay. So it's like building a car versus a spaceship.
Yeah.
Same pieces, different functions.
Yeah. They highlight the diversity that we can get even with the same set of atoms just by changing how they're connected.
That's really cool. Okay, so we've been talking about all these reactions, but I have to admit, I don't really know how they happen on a molecular level. It seems kind of mysterious. Like, how do we even begin to understand what's going on during a reaction?
Well, that's where we get into this fascinating realm of organic reaction mechanisms.
Okay.
So it's really understanding how these reactions occur.
Yeah.
It's crucial for us if we want to predict and control chemical transformations.
Right, yeah, it's like having a blueprint.
Exactly. It's like having that blueprint that lets you build new materials, medicines, all of that.
Cool. Okay, so how do we understand those mechanisms?
Well, first you got to understand how those bonds break.
Huh.
Right. So we have 2 ways, heterolytic cleavage and homolytic cleavage.
Okay, break it down for me. What's the difference?
Heterolytic cleavage, the bond breaks unevenly.
Okay,
One atom keeps both of the shared electrons.
okay.
In homolytic cleavage, it's an even break.
Okay, so it's like splitting a cookie. Sometimes one person gets more, sometimes it's a clean break down the middle.
Exactly.
Cool.
Now, heterolytic cleavage, that gives us ions. Remember those charged species?
Right.
They play a huge role in reactions.
Okay, so walk me through it. What happens with that uneven split?
So let's say carbon loses those shared electrons.
Okay.
It forms what we call carbocation, so it's a positively charged carbon.
Okay.
Now, those carbocations, they're really reactive. They want to fill that electron shell.
So they're looking for electrons to grab.
Exactly.
Oh.
But then, if carbon gains both electrons from the bond cleavage, it forms a carbanion, so that's a negatively charged carbon atom.
Okay, got it.
Carbanions, they're also pretty reactive.
Yeah, that makes sense.
They want to donate those extra electrons.
Okay, so it's like a molecular Robin Hood, taking from the rich, giving to the poor.
That's a great way to put it. Now, homolytic cleavage, that's a bit different. It gives us free radicals.
Okay, free radicals. I've heard of those.
Those are neutral species with an unpaired electron.
Oh, right. So they're super reactive because they're looking for a partner to pair up with.
Exactly.
Okay, so heterolytic makes ions, homolytic makes free radicals. Got it. But how do we know which one happens?
Well, it really depends on the atoms involved and the reaction conditions, but understanding how those bonds break is key for figuring out what happens in the reaction.
That makes sense. So we've got these carbocations, carbanions, free radicals, but how do they, find each other? It seems like it'd be chaos with all these reactive things floating around.
Well, that's where we introduce the concepts of nucleophiles and electrophiles.
Okay, nucleophiles and electrophiles.
So nucleophiles, they're electron-rich.
Okay.
They often have a negative charge or a lone pair of electrons,
Yeah.
and they're attracted to electron-deficient areas.
So they're kind of like those people who always want to be in the middle of the action.
Yeah, they love the nucleus.
Okay, so what about electrophiles?
Electrophiles are the opposite.
Okay.
They're electron deficient,
Okay.
often have a positive charge, and they're looking for electron-rich areas.
Okay.
So they love electrons.
Got it. So it's like a molecular dating service matching up the nucleophiles with their electrophile soulmates.
Exactly. And understanding, which is really key for predicting how a reaction will proceed.
Yeah.
So the nucleophile is going to attack that electrophile, donating its electrons and forming a new bond.
Okay. That makes sense. But how do we actually see these electrons moving around? I mean, they're tiny.
Right. We can't directly see them, but we use something called curved arrow notation.
Okay.
It's like a way to represent those electron shifts. So the arrow shows where a pair of electrons is moving.
Oh, okay.
It starts at the source and points to the destination.
Got it. So it's like drawing a map of the electron's journey as it hops from atom to atom.
That's it.
Okay. But how do we know, which parts of the molecule are more likely to attract those nucleophiles or electrophiles? It seems like some atoms would be more appealing than others.
Yeah, that's a good point. And that brings us to the idea of electron displacement effects.
Okay.
So these effects describe how electrons within a molecule can shift around, and that can make certain regions more attractive to nucleophiles or electrophiles.
So it's like those underground currents that can, change the landscape.
That's a great analogy.
Cool.
And one of the most important ones is the inductive effect.
Inductive effect, okay. What's that?
It's all about how the electron density in a sigma bond can be influenced by the electronegativity of nearby atoms.
Electronegativity, right. That's how much an atom wants to pull electrons towards it.
Exactly. So if you have a really electronegative atom, like a halogen attached to a carbon chain, it's going to pull those electrons toward itself.
Right. It's like a molecular tug of war.
Exactly. And that can create a dipole moment where the carbon has a partial positive charge and the halogen has a partial negative charge.
Okay, I get it. So that inductive effect, can it affect other atoms in the chain?
Yeah, it can actually be transmitted through the chain, so it can influence the reactivity of nearby atoms.
That's interesting. Okay, so the inductive effect is all about those electronegativity differences. Are there other effects that can influence reactivity?
Yeah, we also have the resonance effect, sometimes called the mesomeric effect.
Okay, resonance effect. What's
Yep.
that one do?
This comes in when you've got multiple bonds or lone pairs that can interact through conjugation.
Wait, conjugation, doesn't that have something to do with alternating single and double bonds?
Yes. Conjugation is like a highway for electrons.
A highway, oh.
It lets them delocalize and spread out.
Okay, so the electrons are on a road trip.
Exactly. And when this delocalization involves those pi bonds or lone pairs, that's when we call it the resonance effect.
Gotcha. Okay, so how does that affect reactivity?
Well, the resonance effect can either donate electrons or withdraw them. It just depends on what atoms or groups are involved. So electron-donating groups, like those with lone pairs, they push electron density into the system, making those areas more electron-rich.
Okay.
Electron-withdrawing groups, those often have double or triple bonds to electronegative atoms, and they pull electron density away.
So it's like having two different kinds of travelers on that electron highway. Some are leaving stuff behind and others are taking things away.
I love that analogy. And just like with the inductive effect, this resonance effect can really change how a molecule reacts, making certain spots more or less attractive.
Okay, that makes sense. Are there any temporary electron shifts, like only when a molecule's about to react.
Yeah, actually, that's a great point, and that's called the electromeric effect.
Okay, the electromeric effect. Okay.
It describes how those electrons in a multiple bond move temporarily when something's coming in to react.
Okay, so it's like the molecule's getting ready, like, oh, here comes a nucleophile.
Yeah, exactly, like a molecular mobilization.
That's cool.
The approaching region, that can induce a shift in electron density
Okay.
toward one atom in that bond, making it more susceptible to attack.
So it's like a molecular game of strategy, right? Like, okay, let's position ourselves for this reaction.
Exactly. And then once the reaction's done, the electromeric effect goes away and everything goes back to normal.
Oh, okay, that's interesting. So we've got these bond-breaking nucleophiles, electrophiles, electron displacement effects, all these things working together. But is there anything else we need to know to predict how a molecule might react?
Well, there's one more effect that's really important, especially for understanding how stable carbocations are,
Okay.
and that's hyperconjugation.
Hyperconjugation, that sounds intense.
It is kind of cool. It's basically a stabilizing interaction that involves those sigma electrons
Okay.
from a CH bond that's right next to a carbocation.
Okay.
Those sigma electrons can kind of overlap a little bit with the empty p orbital of the carbocation,
Okay.
and that helps spread out that positive charge, making it more stable.
So it's like the carbocation is getting a little support from his neighbors.
Exactly. And the more CH bonds there are nearby, the more stable that carbocation is.
Okay, that's cool. So all of this is really fascinating, all these different effects, but it seems like understanding them is super important for chemists.
Right. Absolutely. Yeah. I mean, knowing how reactions happen, it allows us to predict the products. We can control the conditions to get specific products.
Oh, wow.
And we can even design new ways to make molecules with properties we want.
So it's like you have this toolbox
Yeah.
with all the knowledge, and you can build anything.
Exactly. A molecular toolbox.
I love it. Okay, but with all this talk about molecules and reactions, I'm curious what it's actually like in an organic chemistry lab. What kind of tools and techniques do you use to do all this?
Oh, that's a great question. It's a world of creativity and precision.
Okay, I'm ready to put on my lab coat and goggles. What are some of the essentials?
Well, one of the most basic but essential is glassware. We use flasks, beakers, condensers, all sorts of things to contain and work with the reactions.
So it's like your kitchen, but for molecules.
Exactly. And just like in a kitchen, keeping everything clean is super important.
Yeah, that makes sense.
Even a tiny bit of impurity can mess up an experiment. We clean and dry everything meticulously.
Okay, so precision is key.
Absolutely. But beyond the glassware, we also have a lot of specialized equipment. One of the most important is a heating mantle or a hot plate.
Okay, so that's like the stovetop in our molecular kitchen.
Exactly. It helps us heat the reactions to specific temperatures.
Okay.
And to make sure things heat evenly, we often use a stir plate with the magnetic stir bar.
Oh, okay, to keep things mixed up.
Exactly. It's like our molecular blender.
Nice.
And of course, we need to monitor the temperature. We use a thermometer or thermocouple for that.
Okay, so it's like the oven thermometer, making sure we don't burn anything.
That's a good way to think about it. Now, sometimes we need to cool reactions down too. We use ice baths or cooling baths for that.
Okay, so we've got the fridge in our molecular kitchen too.
Exactly. And then to separate solids from liquids, we use filtration with filter paper and a funnel.
Okay, so that's like our strainer,
Yeah.
separating the solids from the liquids.
You got it. And to get rid of solvents or purify liquids, we use something called a rotary evaporator, or rotovap for short.
A rotovap, okay. What's that?
It uses a vacuum to gently remove those solvents,
Okay.
leaving behind what we want.
So it's like our molecular dehydrator.
Exactly. And once we have that purified product, we need to analyze it, make sure it's what we think it is.
Right, yeah, how do you do that?
For that, we use things like spectroscopy and chromatography.
Ah, right, so now we get to be molecular detectives.
Exactly.
Okay, so spectroscopy and chromatography, can you remind me what those are all about?
Sure. Spectroscopy is like a way for us to see molecules. We look at how they interact with light or electromagnetic radiation.
Okay. There are different types like infrared spectroscopy and nuclear magnetic resonance spectroscopy.
Okay.
They give us different clues about the molecule's structure and its functional groups.
Okay, got it. So what about chromatography?
Chromatography, that's a technique we use to separate mixtures of compounds.
Okay.
Based on how they interact with a stationary phase and a mobile phase. It's like a molecular obstacle course, where different compounds move at different speeds depending on their interactions with the stationary and mobile phases.
So we're using light to see molecules with spectroscopy, and we're separating mixtures with chromatography. It sounds like organic chemists have a whole toolkit for analyzing those molecular mysteries. But I have to admit, thinking about working with potentially hazardous chemicals makes me a bit apprehensive.
Mhm.
What safety precautions do organic chemists take to protect themselves in the lab? I mean, those beakers and flasks don't exactly scream safety first, do they?
That's a really good point. Safety is the most important thing in an organic chemistry lab.
Okay, good. We always wear the proper personal protective equipment, so lab coats, gloves, eye protection,
Okay.
to protect ourselves from the chemicals. And we work in fume hoods,
Fume hoods, right?
those special ventilation systems.
Yeah.
They pull away any harmful vapors, like a superpowered exhaust fan.
So we're talking molecular armor and molecular exhaust systems. It sounds like you're taking every precaution to stay safe in that molecular playground.
Yeah, we take safety very seriously. We handle all those chemicals really carefully,
Okay.
following specific procedures
Okay.
for measuring, transferring, disposing of them.
That's good to know. Okay. Well, this is all really fascinating, but I'm starting to feel a little overwhelmed, to be honest.
Yeah, it can be a lot.
It's a lot. Is there a way to simplify all of this? Like, are there any, underlying principles that can help us organize all this information?
Yeah, for sure. One of the most helpful things to remember is those functional groups.
Right. Those molecular tags.
Exactly. If you know what functional groups are in a molecule, you can start to predict how it'll react,
Okay.
what kind of products you might get. So, for example, if you see a hydroxyl group, you know you've got an alcohol.
Yeah?
And then you can start thinking about reactions like dehydration or oxidation.
Ah, so it's like having a little cheat sheet.
Yeah, exactly.
Okay, cool. Are there other organizing principles like those functional groups that can help us?
Yeah, definitely. There's also the idea of reaction mechanisms.
Right, those step-by-step descriptions of how things happen.
Exactly. So once you understand the mechanisms of some key reactions, you start to see patterns, and you can predict how similar reactions might go.
So it's like having the playbook, understanding the moves those molecules are making.
That's a great way to put it.
Okay. But how do you even figure out what's in a compound? Like, you can't just look at it and know.
Right. You're right.
Okay.
We
good.
can't just see the atoms and count them.
All right.
Determining the composition of a compound, it's really important. And it involves both qualitative and quantitative analysis.
Right. Qualitative and quantitative. Can you remind me what those mean?
Yeah, so qualitative analysis is like detective work.
Okay.
You're trying to figure out what's there.
Okay.
What elements, what functional groups. Quantitative analysis, that's figuring out how much of each component is there.
Okay, so qualitative is like what, and quantitative is like how much. Got it.
Exactly.
So how do you figure out what elements are in an organic compound?
Well, first we want to make sure it's actually organic.
Right. It has to have carbon.
Exactly. One classic test, we heat it up really strong.
Okay.
If it turns black and chars, leaving carbon behind, that's a good sign.
So it's like you're barbecuing the molecule.
Yeah, something like that. Of course, we also have more sophisticated techniques like combustion analysis,
Okay, combustion analysis.
where we burn the compound very carefully
Okay.
and look at the products to see how much carbon, hydrogen, and other elements are there.
So that's the quantitative part. But how do you even know if a compound has nitrogen or sulfur or halogens?
For those, we use a test called Lassane's test.
Okay, Lane's test.
It involves fusing the compound with sodium metal,
Okay.
and that transforms those elements into ionic forms.
Oh,
So it's like we're giving those elements a makeover
that's a great way to put it.
so we can detect them. So, for example, if we add iron 2 sulfate and acid to the extract and we get a deep blue color,
Okay.
that means there's nitrogen.
Oh, wow, that's so cool. So it's like a molecular mood ring.
I like that. And we have other tests for halogen, sulfur, phosphorus.
Okay. So once you've done all that detective work, how do you actually measure the percentages of each element?
For carbon and hydrogen, we use that combustion analysis.
Right.
We burn it in oxygen,
Okay.
and then we carefully measure the masses of carbon dioxide and water that are produced.
So it's like a molecular weight loss program.
Yeah, and from those masses, we can figure out the percentages.
Cool. Okay. What about nitrogen?
For nitrogen, we have a couple of options. One is the Dumas method, where we combust the compound and measure the volume of nitrogen gas.
Okay.
Another is the Kjeldahl method,
Okay.
where we convert the nitrogen to ammonia, and then we titrate that with an acid to figure out how much there is.
Wow. It's amazing how many different techniques there are for all of this. Okay. So what about oxygen? How do you measure that?
Oxygen is a bit trickier. A lot of times we actually just determine it indirectly
Okay.
by subtracting the percentages of all the other elements from 100%.
So it's like a molecular game of hide and seek.
Exactly.
Okay, that's clever.
Hmm.
Okay, so once you have those percentages, how do you get the actual formula of the compound?
That's where we use something called the empirical formula.
Okay, the empirical formula. Okay.
It's the simplest whole number ratio of the atoms in the compound.
So it's like the most basic recipe.
Exactly. And from the empirical formula, we can often get the molecular formula, which tells us the actual number of atoms of each element. So it's like going from the basic model to the deluxe version.
Right, getting the full blueprint.
Yeah.
But how do you go from percentages to formulas?
It does involve a bit of math.
Okay.
You have to convert those percentages to masses,
Okay.
then the masses to moles,
Okay.
and then find the simplest ratio of those moles.
Okay, I remember doing those calculations back in chemistry class.
Yeah, it's a bit of stoichiometry magic.
Okay, but once you have the molecular formula, how do you know the actual structure? I mean, you could arrange those atoms in different ways, right?
Yeah, absolutely. And that's where our knowledge of bonding and functional groups and isomerism comes in.
Right. Okay.
We use all of that, along with information from things like spectroscopy, to figure out the most likely structure.
Right, spectroscopy. You mentioned that earlier. Can you remind me how that helps us see the structures?
Sure. So spectroscopy, it analyzes how a molecule interacts with light or electromagnetic radiation.
Okay.
Different types of spectroscopy give us different information. Infrared spectroscopy tells us about the bonds.
Okay.
NMR, nuclear magnetic resonance spectroscopy, tells us about how the atoms are connected and their environment.
Okay, so it's like using different lenses to get a clearer picture.
Exactly. And then we have mass spectrometry.
Right, mass spectrometry, okay.
That one helps us figure out the molecular weight of the compound and how it breaks apart.
Okay,
So that gives us more clues about its structure.
so we're weighing those molecules and then smashing them to see what they're made of.
Exactly. It's like a molecular demolition derby.
I love it. Okay, well, all of these techniques are really impressive, but I have to ask, what's the point of all this?
Oh, that's a good question.
Like, why do we care about all these organic molecules and reactions? It's super interesting, but is it useful?
It's definitely useful.
Okay.
Organic chemistry is not just some, academic thing.
Yeah.
It impacts almost every part of our lives.
Really? Okay, how so?
Well, one of the biggest areas is pharmaceuticals.
Oh, right, medicines.
Almost all drugs are organic compounds.
Okay.
They're designed to interact with certain targets in the body.
Right, to treat diseases.
Exactly. So organic chemists were the ones making those drugs.
Oh, wow, that's a big responsibility.
Mhm.
What other areas use organic chemistry?
Another big one is agriculture.
Agriculture, okay.
So pesticides, herbicides, fertilizers, those are often organic compounds
Okay.
designed to protect crops, provide nutrients.
Okay, so organic chemistry is helping us grow our food. That's important.
Absolutely. And then there's materials science.
Material science.
Plastics, fabrics, all sorts of things are made from organic polymers.
So it's like a molecular construction set, building all the materials we use every day.
Yeah, exactly. We design those polymers to have specific properties
Okay.
to do what we need them to do.
Wow. It's incredible how these tiny molecules make up all these things we see and use all the time. Are there other fields that use organic chemistry?
Oh, yeah, for sure.
Okay.
Energy is another big one.
Oh, right.
Yeah.
Fossil fuels.
Yeah, those are made of organic compounds. But now we're also using organic chemistry to develop new energy sources.
Okay.
Things like biofuels, solar cells, better batteries.
So it's like organic chemistry is helping us power the world.
Exactly.
This is all really amazing. It seems like organic chemistry is everywhere.
It really is. It's essential to so many things.
Yeah. Okay, so we've talked a lot about the science, but I'm curious about what it's like to actually be an organic chemist.
Mm.
What's it like to work in this world of molecules?
It's really exciting.
Yeah.
For me, it's the challenge and the creativity. It's like solving a puzzle,
Okay.
Figuring out how things work at that tiny level.
Yeah.
And then getting to make something new, designing a molecule that's never existed before.
Wow, that's really cool. But I'm sure it's not always easy.
Oh, definitely not.
I mean, do those reactions always work perfectly?
Of course not.
Okay.
There are always surprises.
Okay.
Reactions take unexpected turns. You get weird byproducts. It can be frustrating, but it's also fun. You learn something new every time.
So you got to be pretty resilient to be an organic chemist.
Yeah, that helps.
Okay, so what advice would you give to someone who's interested in organic chemistry? Where do they start?
Well, first I'd say get a good foundation in the basics.
Okay. The basics of chemistry.
Yeah, atomic structure, bonding, stoichiometry.
Right.
And then from there, dive into the world of organic molecules.
Okay.
Learn about functional groups, reaction mechanisms, those spectroscopic techniques we talked about.
Okay, so it's kind of like learning a new language, the language of molecules.
Exactly. And then, of course, you need to get some hands-on experience in the lab.
Right. That's where the magic happens.
Exactly. It's a challenging field.
Yeah.
But it's so rewarding. You never know. You might be the one who discovers a new drug or designs a new material or comes up with a solution to the energy crisis.
Wow, that's really inspiring. Okay, well, this deep dive into organic chemistry has been amazing. I've learned so much, and I feel way more informed now. Thank you so much for sharing your knowledge with us.
Oh, it's my pleasure. Really, it's great to talk about this stuff. And remember, it's a huge field, and it's always changing, so keep learning and exploring.
That's great advice.
It really is a fascinating world.
It is.
Mm-hmm.
But as we've been talking, it's kind of hit me that, like, even a tiny change in a molecule can make a huge difference in what it does.
You're absolutely right. And that brings us to one of the most, I think, fascinating and sometimes mind-boggling parts of organic chemistry, isomerism.
Isomerism. Yeah, we
Yeah.
talked about that a little bit before with the Lego analogy.
Right.
But can we, like, dive a little deeper into that? What exactly are isomers, and why are they so important?
So isomers are compounds. They have the same molecular formula, same number and types of atoms, but the atoms are arranged differently.
Okay.
It's like having two sets of Legos, same exact pieces,
Right.
but you build a car with one and then like a spaceship with the other.
I like that.
Same pieces, but totally different structures and functions.
Okay, yeah, that visual really helps. So two isomers might have the same formula, like C4H10, but they're different because those atoms are arranged in different ways.
Exactly.
Okay.
And there are a few types of isomers, each with their own, little quirks. We can break them down into structural isomers and stereoisomers.
Okay, structural isomers and stereoisomers.
Yeah.
Let's start with structural isomers. What are those?
So structural isomers have the same atoms, but they're connected differently.
Okay.
Back to the Legos, you can arrange the same bricks in tons of ways.
Right.
With structural isomers, it's like we're rearranging the bonds between those atoms.
Okay, so it's all about how they're linked together.
Exactly. And there are a few types of structural isomers, too.
Okay, I'm ready. What are they?
Well, we have chain isomerism,
Okay.
where the carbon backbone, those Lego chains,
Yeah.
can have different branching patterns. For example, butane and isobutane, they're chain isomers.
Okay, butane and isobutane.
Butane's a straight chain of 4 carbons. Isobutane has a 3-carbon chain with a methyl group branching off.
So like a straight Lego chain versus a branched one.
Exactly. And even though they have the same formula, they have different properties.
Okay.
Like their boiling points are different.
Wow, that's so cool. Okay, what other kinds are there?
We also have position isomerism,
Position isomerism. Okay.
where a functional group or a substituent, one of those Lego side groups,
Yeah.
it's attached to a different carbon on the same chain.
Okay.
For example, 1-propanol and 2-propanol.
Okay.
They both have a 3-carbon chain, but the hydroxyl group's in a different spot.
So like moving that hydroxyl group Lego piece to a different spot on the chain.
Exactly.
Okay.
And again, even though they have the same formula, those little changes can make them react differently.
That's so cool. Okay, chain isomers, position isomers. What else? What other structural isomers are out there?
We also have functional group isomerism.
Functional group isomerism. Okay.
That's where the same atoms make different functional groups.
Oh, okay. So we're switching out those molecular tags, giving them different personalities.
Exactly. A classic example is ethanol and dimethyl ether.
Okay.
Both have the formula C2H6O,
Okay.
but ethanol's an alcohol, dimethyl ether's an ether.
Wow. Completely different compounds. It's amazing how just changing how they're arranged makes such a big difference.
Yeah, it is. And these are just the structural isomers. We also have stereoisomers, which make things even more interesting.
Okay, stereoisomers. Right. We were talking about mirror images before. Can you remind me how those fit in?
Yeah, so with stereoisomers, the atoms are connected in the same order,
Okay.
but they're arranged differently in space. It's like those Lego models that are mirror images.
Right.
They have the same pieces, same connections, but they don't quite line up.
Yeah, it's like your left and right hands, they're mirror images, but you can't perfectly stack them on top of each other.
Exactly. And that handedness, we call it chirality.
Chirality, okay.
It's really important in biological systems. Lots of molecules in our bodies, like enzymes, receptors, they're chiral. And just like your left hand fits better in a left-handed glove, one version of a drug might work better than its mirror image.
Wow, that's so cool. So chirality actually matters. Okay, so What are some examples of those stereoisomers?
One type is geometric isomerism, also called cis-trans isomerism.
Okay.
It happens when you have restricted rotation around a double bond, like in alkenes.
Right, because of those pi bonds.
Exactly. If two groups are on the same side of the double bond, it's cis.
Okay.
If they're on opposite sides, it's trans.
So like those Lego side groups are facing the same way or opposite ways.
You got it. And there's different orientations. They can change the molecule's shape and how it reacts.
Okay, that makes sense. So geometric isomers, those are cool. What other kinds of stereoisomers are there?
We also have optical isomerism.
Okay, optical isomerism.
That comes from a chiral center.
A chiral center, what's that?
It's a carbon atom with 4 different groups attached.
Oh, okay, so it's like that carbon is the hub, and each of its bonds has a different Lego piece attached.
Exactly. And because of those 4 different groups, that carbon can exist in 2 different arrangements that are mirror images. We call those enantiomers.
Okay, so those are the non-superimposable mirror images.
Yeah.
Like our hands.
Exactly.
Got it. It's amazing how much variety there is in these molecules, but honestly, I feel like we've only just scratched the surface.
You're absolutely right. Organic chemistry is a huge field, always growing and changing.
Yeah. But this deep dive has been incredible. I've learned so much, and I feel so much more like aware of this molecular world around us. Thank you so much for being such a great guide through all of this.
Oh, you're very welcome. It's been my pleasure. And just remember, always keep asking questions, keep exploring, and never stop being curious about the world. Who knows what amazing discoveries are out there waiting to be made?
That's great advice. And to all our listeners, thanks for joining us on this deep dive into organic chemistry. We'll see you next time.